In this activity, you will examine plants whose leaves are modified to capture and digest animals. Carnivorous plants typically grow in environments such as bogs and wetlands, where the soil is low in essential nutrients, especially nitrogen. To survive, these plants have evolved leaves that function as traps, allowing them to obtain nutrients from insects and other small organisms. Carnivorous plants are most commonly found in habitats like bogs, fens, marshes, and other wetland environments. These areas usually have plenty of water and sunlight, but the soil is extremely poor in nutrients, especially nitrogen and phosphorus. Because the soils are often acidic and low in oxygen, decomposition happens very slowly. That means nutrients are not released back into the soil the way they are in more typical ecosystems. So even though these plants have water and light for photosynthesis, their roots can't obtain enough of the minerals they need for growth. To survive in these nutrient-poor conditions, some plants have evolved a very different strategy. Instead of relying only on their roots, they use their leaves to capture and digest insects and other small organisms. The nutrients released from this prey, especially nitrogen, supplement what the soil cannot provide. So carnivorous plants are not eating insects for energy. They still get their energy from photosynthesis. They capture insects to meet their nutrient needs, so protein, not carbs, allowing them to thrive in environments where most other plants would struggle. Carnivorous plant traps fall into two main categories. Active traps use movement to capture prey. Passive traps rely on structural features that prevent prey from escaping. Both types are formed from highly modified leaves. Active traps use movement or rapid physiological responses to capture prey. The Venus flytrap has hinged leaf lobes with sensitive trigger hairs. When these hairs are touched, the leaf snaps shut, enclosing the insect and releasing digestive enzymes. Sundews have leaves covered with glandular hairs that secrete sticky droplets. When an insect lands, it becomes trapped in the adhesive, and the leaf slowly curls around it to increase contact with digestive enzymes. Bladderworts are aquatic or semi-aquatic carnivorous plants with tiny bladder-like traps on their leaves. These bladders function as suction devices. When small aquatic organisms touch trigger hairs near the opening, the trapdoor opens. Water and prey are rapidly sucked in, and the door closes again in milliseconds. Digestive enzymes then break down the captured organism. These traps are considered active because they involve rapid movement and pressure changes. Passive traps capture prey using specialized shapes and surfaces rather than rapid movement. Pitcher plants use pitfall traps. Their leaves form deep, fluid-filled chambers with slippery rims and downward pointing hairs. Insects fall in and are unable to climb back out, eventually being digested. Lobster pot traps, found in plants such as Gynlecia, corkscrew plants, use inward pointing hairs that guide small organisms into a tubular chamber. The hairs are arranged like the entrance to a lobster trap, easy to enter but difficult to escape. Once again, prey are directed toward digestive regions of the leaf. These traps rely entirely on structural design and directional guidance rather than movement to capture and retain prey. Before observing the specimens, think about the relationship between nutrient availability and plant survival. Why have certain plants evolved leaves that capture and digest insects instead of relying solely on photosynthesis? Consider what resources are limited in bog environments and how carnivory compensates for those limitations. Record your prediction in your lab notebook before continuing. Observe each carnivorous plant specimen provided. This one is sundew. In your lab notebook, sketch the modified leaf and label the name of the plant, whether the trap is active or passive, and key structural features involved in prey capture and digestion. Focus on how the shape and surface features of the leaf relate to its trapping mechanism. Pause the video to sketch the plant. This is the pitcher plant. Notice how the pitcher is an extension of the green leaves where it is connected by a tendril. In your lab notebook, sketch the modified leaf and label the name of the plant, whether the trap is active or passive, and key structural features involved in prey capture and digestion. Pause the video to sketch the plant. This is a Venus flytrap. Notice that the petiole has been modified into a phyllodes, which appears leafy. This increases photosynthesis in the plant. In your lab notebook, sketch the modified leaf and label the name of the plant, whether the trap is active or passive, and key structural features involved in prey capture and digestion. Pause the video to sketch the plant. Answer the following compare and contrast questions. First, how do leaf shape and structure relate to the trapping and digestion mechanisms? Connect movement, stickiness, or pit structure to how prey is captured and retained. Next, what structural features are shared by all leaves despite their different functions? Think back to the epidermis, mesophyll, and vascular tissue. Even in highly modified leaves, these basic tissues are still present. Record your responses in your lab notebook. Whether snapping shut, secreting sticky mucilage, generating suction, or forming pitfall traps, carnivorous plant leaves illustrate how a single organ type can involve extraordinary specializations. Despite these differences, all still contain epidermis, mesophyll, and vascular tissues, reminding us that even the most dramatic adaptations are built upon the same fundamental plant structures.